So it's a great honor to kick off the meeting right now with our first keynote speaker, Dr. Susan Gasser. Susan was a group leader at the FMI. I won't try to pronounce the name in German, until 2021, which she actually directed it until 2019. She was also a professor at the University of Geneva at the same time. And since 2021, she went on to go to be the director of the ICERC Foundation, a foundation that supports translational research in cancer. So Susan has a very long history of seminal findings. It's an honor to have her here. She identified topoisomerase II as a structural component of the mitotic chromosomes, IT-rich sequences as organizers of loop organizations. She moved forward a lot of understanding on mechanisms that tether chromosomes to the nuclear envelope, and many of the mechanisms that are responsible for genome integrity. She has authored more than 300 papers, received too many awards for me to list today. She has held honorary positions, and she has advised governmental bodies on top of all that, she even had the time to promote and advocate for women in science. So it's really a role model for me, and a real pleasure to have her here. Very much looking forward to your talk.
Thank you. So it's my first time at this meeting, though I did know it was going on and it looked extremely interesting. So I'm not really going to talk about epigenomics per se. I'm going to talk about nuclear organization. That's what I've worked on basically all my career. And the question is how that impacts health and disease. So one thing I think we'll all agree with is that basically the differentiation from a pluripotent progenitor cell to a committed precursor, and then maintenance of this state and final differentiation, it is the basis of life, of complex organisms. And maintenance of that, since they all have the same genome, no matter what they turn into, either maintenance of a pluripotent or a committed state, a memory of what that cell should become, is epigenetic. Now, coupled with that epigenetic changes or restriction is a change in the organization of the genome. In an ES cell, the nucleus is rather nondescript, I would say. It looks rather uniform. You don't see much dense. You can always identify nucleally, but not much else. Whereas in differentiated cells, you increasingly accumulate very dark staining, negative stained chromatin. Question is the spatial segregation, and this, of course, is a segregation of heterochromatin and euchromatin. Question really is that spatial segregation part of the mechanism that maintains and contributes to cell type identity and then integrity? We're going to have the lights down on the stage because I have lots of fluorescence. So could someone turn off the lights? You don't have to see me, but you do have to see the slides. Okay, so there are many ways to look at the nucleus and study it. There's the organization of chromosomes. There's the organization of the individual fiber. You can look at single molecule transcription, track single molecules and RNAs and DNA in time, look at transcription foci. But one tool that has been extremely useful has been to look at chromatin modifications and their compaction into repressive compartments. This is true in every eukaryote, from budding yeast, that's where I started my career, telomeric foci up through pericentric polycomb and the general heterochromatin that organizes the repressed domains. So this is, the question is what really is what's the proof? What is in these compartments? What's the evidence that it's compacted? Now, one main message I want to give you is that, in fact, the organization of the chromatin from the very individual fiber with the histone modifications up through the whole organization, the nucleus, is really driven by histone modifications. So, in fact, epigenetic modifications are somehow, as modest as they may seem, and because they're molecular size, they probably are driving to a large part the global structure, whether it's into TADs, active and inactive domains, compartments of early and late replication, or whole chromosomal positioning and segregation. So what is in these repressive domains at the nuclear periphery? Well, it's not negative staining, actually, does not tell us anything. It actually isn't even quantitative. But David Bassett-Jones did a beautiful job in the early 90s with electron spectroscopic imaging, where he could quantitatively measure phosphate and nitrogen content on micrographs. And in differentiated nuclei, this is a liver, and this one's a lymphocyte, he could show the density of phosphate was much higher in the heterochromatic or the repressed domains. Also nitrogen, when you merge, you see this beautiful overlap, up to 10-fold more dense than in the euchromatic parts. And this, of course, is enhanced in various nuclei where there's an increasing amount of heterochromatin. Later with CLEM and antibodies, this could be correlated with H3K9 methylation and H3K27 methylation, the two classic repressive marks. So, textbook view, of course, open chromatin is acetylated, more accessible, transcribed often. There's a polycomb repressed chromatin and H3K9 methylation repressed chromatin, both sequestered into foci or some kind of compartment. But it's not just repetitive elements, and so actually it isn't really just constitutive heterochromatin that is organized by K9 methylation, but also a large number of tissue-specific genes in tissues where those genes need to be off. So naturally, tissue-specific genes will have a proper tissue in which they're on, but in all others, then they should be sequestered away. And in fact, much of that is mediated by K9 methylation. Now, in mammals, invertebrates, there is a DNA methylation, but it is actually usually secondary to histone methylation. And since I'll be talking about C. elegans to a large part, we're going to focus on the histone methylation, not the DNA methylation. And then instrumental in the DNA methylation and control of it, and then, of course, control of the integrity of the genome, the genomic pattern, are the histone methyltransferases, or HMTs, that deposit sequentially monodi, and then trimethylation on lysine 9, or in the case of polycomb, on lysine 27. So why are we so interested in repeats and repression? Well, if we look at the genome over time, and there's about a 300-fold increase in genome size from yeast, budding yeast, to man, but only less than a fourfold, 3.5-fold increase in number of genes. So there is a discrepancy here. And we can look at the halfway house, the C. elegans, where there are about 20,000 genes, 10 times more genome. And the only thing that really, really expands is repeats. The more complex the organism, the more the accumulation of simple satellite repeats, but also, of course, transposons, retrotransposons, DNA transposons, and then intervening non-transcribed or transcribed or non-coding regions. So humans deal with that. We have to deal with that. But we're really talking, when we talk about the coding genome, it's really, really little. And then there's a massive amount of baggage we have to take care of and make sure that if it can be used, okay, it may be used at some times, but how do we deal with this? Now, the repeats fall into 4 classes, and this is sort of a classic from a review we wrote. But anyway, the largest class is, of course, transposable elements. These are the viral elements that invaded and now became part of our genome. There are DNA transposons, LTR retrotransposons, non-LTR retrotransposons like the line elements, which in primates certainly make the vast majority, and sign elements. The interesting thing is that all of these have Pol II sites in their terminal repeat, and these, of course, in number, far outnumber the number of Pol II sites on genes. So it's very important to realize that if these were not sequestered away, they would be mopping up, in fact, the majority of our Pol II. So they are heterochromatic, and most of them carry H3K9 or trimethyl. Then there are telomeric repeats, the TG repeat. There's flanking the centromere, large, vast expanses of 170 base pairs, centromeric satellite. There are microsatellites and mini satellites ranging from 2 to 5 up to 30 base pairs of simple repeat scattered through the chromosome arms. In any case, this is part and parcel of our genome, and as I will show you, I hope to convince you by the end of the day, keeping it in, under control is essential for health. Now, the HMTs then become the agents of control. The HMTs, of course, are the histone methyltransferase, highly conserved enzymes. There are 6 in mouse and man, in most mammals. They have a very structurally conserved catalytic set domain with a pre and post, conserved pre and post set domain motifs. They fall into families. The SUVAR39 was originally identified in flies as being the mediator of PEV from centromeric repeats. There are 2 very similar, both with chromodomains in human and mouse. SETDB1 is a very important, and I'll talk about the worm homologue of this almost the whole talk. Very important histone methyltransferase, is in embryonic lethal, identified in mouse. And then G9A and GLP were identified by Shinkai of Tokyo, and he found that at telomeres as a component of telomeric chromatin, heterochromatin, and it also has ankyrin repeats, just as the others each have important repeats that either recognize DNA histone marks or mediate interaction with other proteins. Now, worms only have 2. They have a very clear SETDB1 homologue, which we would call MET2. On all my slides, I'll also use SETDB1 so that you don't get lost because of C. elegans names. Has all the structure features and the same partners. And then SET25, which has a set domain that's very similar to G9A and SUVAR39, but it doesn't have any of the bells and whistles that usually go with it, and has a different role. So now worms, as long as I introduced our organism, its genome is about 30-fold smaller than, or 10-fold smaller than human. It's got all the transposable elements in different ratios, so it has far fewer line elements, still has a lot of non-LTR retrotransposons, but has a lot more DNA transposons. Why it has DNA rather than RNA transposons, I don't know, but that's the case. Somebody may know. It has telomeric repeats and micro and mini satellites spread along the arms, but it doesn't have centromeric satellite because it has something that is a satellite repeat spread along the chromosomal arms, but not a centromeric satellite because it's holocentric. That means actually the microtubule can attach anywhere along the chromosome arm and doesn't need a surrounding cushion of centromeric satellite DNA. So holocentric chromosomes mean that actually chromosome segregation in worms is not dependent on H3K9 methylation, which normally organizes the centromeric satellite. which is a big advantage because we're never looking at a chromosome loss phenotype when we look at loss of H3K9 in worms. Now, we still have all these Pol II sites that outnumber the genes. And the other thing that's interesting is in worms, the transposons, then their integration tend not to be in very, very large, long, megabase long arrays. Rather, they are discrete. Usually you can map transcripts from individual retrotransposons, which you can't do easily in mammals. So this is a big advantage. There are about 70,000 mappable repeats. So we could do ChIP-seq, we could do RNA-seq, we could do all these things to look at specific repeat control. And all are coded with K9 methylation. So the question is, what happens if we repress them? Okay, what are the phenotypes? What happens? So we're using C. elegans. So we're making a double knockout. If we knock out both of them, these are the mammalian names. You do mass spec on histones. There's no longer any detectable H3K9 methyls. The same thing is true by immunofluorescence. We see that if you knock out just Set25, MET2 is sufficient to do mono and di. On K9, there are no other methylation marks that are changed. And so we can say that actually they do work sequentially. MET2 does mono di. Often Set25 will add the tri, but not always. And on simple repeats, in fact, dimethyl is sufficient to repress. Actually, dimethylation represses equally well as trimethylation. So what are the physiological repercussions of the double knockout? Just to put it aside, there has been just recently, or last year, a heptuple knockout in mouse cells by Thomas Genuine, who was one of the discoverers of the set domain. And it is lethal, and you can see total loss of H3K9 in tissue culture cells. But of course, this is not you can't study this in a multicellular organism, nor can you ask how that affects, physiological life of an organism because it's embryonic lethal. So the worms actually grow without H3K9 methyl. This was our first shocker in a sense because, it's a multicellular organism. It has all the tissues, gut, muscle, nerves, skin, reproductive organ, etc. And all these tissues differentiate without H3K9 methylation. So that was interesting, but then when we scored them, we found some interesting phenotypes. First, derepression. So here, blue is derepression, log 2-fold change. So these are very, very heavily derepressed. And repeats are in particular heavily derepressed. Tandem repeats, these are just arrays of simple sequence that we can map to different parts of the genome and find them in RNAs that normally are never expressed. So there's a strong derepression of simple repeats, of RNA transposons, and of DNA transposons, both in germline and somatic tissues. There are also genes that are derepressed. There are about 350 of the 20,000 genes are 2-fold derepressed reproducibly in embryos. We're looking in embryos here, mixed stage embryos. And all those that were derepressed actually carried H3K9 methyl in the wild type, so they are direct derepression events. But it's really only a small fraction, about 20%, a fifth of all the H3K9 methyl marked genes that are actually derepressed, because perhaps in these embryos there aren't the appropriate transcription factors. Still, it's a significant correlation. What happens physiologically, even though you can create a worm, that worm has problems. So the germline, which the vulva and the germ cells that develop in this hermaphrodite, and in the hermaphroditic gonad, accumulate DNA damage massively. So we're mapping an apoptotic marker that's a cell surface marker, and you see that there's a big increase in the double mutant, but even massive increase if you increase temperature slightly, which means then that germline cells are actually dying. And in fact, that manifests itself that the progeny number drops dramatically. So the double mutant, no K9, in 2 generations, there's no progeny. So if we grow it, and I can't explain this exactly why, but if you grow it at low temperature, they have fewer progeny, but they still can reproduce. But at 20 and 25, which of course is the natural temperature in which they live, since they live in the soil, they die. So what is this DNA damage? We sequence, etc. It's both insertions and deletions in repeats particular. It's amplifications, so copy number variation of tandem repeats, transposons, anything that was actually K9 methylation normally, and transpositions to a certain extent, transpositions of retroviral elements. So in fact, there is a massive, if we look at worms, we use a colorimetric assay. Basically in adult worm, every tissue has some sort of reorganization and mutation. It's a massive accumulation. So I'm not going to bore you with exactly what MET2 does and CET25, but we spent years then mapping, which one deposited which mark on which sequence, so genome-wide ChIP-seq and RNA-seq in both single mutants and double mutants. And it's all summed up here. The CETDB1 homolog, MET2, which can only do dimethyl, targets all the tandem repeats and many DNA transposons, and that is its unique job. CET25 can't do it. It's upstream of CET25 for tissue-specific genes in the tissues where they're repressed. And it's really the workhorse. It's expressed in all somatic tissues quite significantly. It's required for somatic maintenance of heterochromatin, and its loss promotes infertility, aging, and loss of tissue integrity. And I'll come back to aging. CET25 does target tissue-specific genes and pseudogene, but if you knock it out, the MET2, which deposited the dimethyl, is still sufficient to repress. But what is unique to CET25 is that it alone can target new retrotransposons. So in a sense, this is the security blanket against new viral insertions or retroviral insertions. And it is able to be targeted by an RNA transcript and an argonaut to repress de novo newly integrated DNA. It's high in germline cells, and its loss affects transgeneration repression and retrotransposon repression. So what was really striking to us was this MET2 was de-repressing uniquely or strongly simple repeats as well as genes. And if you look at, this is the MET2 versus the CET25 single mutant before I was showing you the double. CET25 clearly affects certain transposons very, very strongly, and some of these are the most abundant transposons. But MET2 clearly has this unique role in simple repeats. What happens when you express simple repeats? We found that the worms become very sensitive, besides becoming sterile or losing fertility and having germline apoptosis, they become very hypersensitive to HU, which is an inhibitor of replication. They accumulate replication stress, replication damage, and if you probe for something that we call R loops, which where an RNA hybridizes to the DNA and somehow prevents the replication fork progression, we see a massive accumulation in MET2 cells, just as in the double mutant. These spots here, these are embryos. Each one of these bright clusters here is an embryonic nucleus, and you see in the nucleus dozens of R loops, aberrant structures that are, we think, engendering this replication fork damage. Now, we can map these R loops by doing a kind of precipitation and sequencing, and they map to promiscuously transcribed repeats, mostly simple repeats or DNA transposons, or this abundant satellite repeat, and not to retrotransposons that MET2 does not silence. So it really seems that promiscuous transcription leads to a lot of DNA damage. We then did a synthetic lethal screen with MET2 and with a little bit with and without HU, but even without HU, we found it was synthetic lethal or basically almost synthetic lethal with a whole cluster of genes that are required for replication fork stability. And you notice these are, I think I put the mammalian names, but these are tumor suppressors like BRCA1, ATR, BARD1 is the BRCA partner, SMARCA, which is a recombination associated or fork restart associated remodeler, RAD51, and so forth. This is a really striking group of genes that we know because I worked on it, but so it popped out for me, but also the program identified it as fork damage and recovery. Now, we could show that actually this apoptosis of germ cells was completely mappable to MET2 and not SET25, and that it was p53 dependent, so which is the DNA damage checkpoint pathway. Now, the upshot of accumulating a lot of DNA damage and not being able to replicate your genome efficiently is actually, I think it's leaked, is a shorter lifespan. So MET2 worms, just mutation of MET2 alone, shortens lifespan by more than 10%, about 15%. And it really is due to the MET2 because we can suppress it with a demethylase inhibitor or extend lifespan with a demethylase inhibitor. And so this is another aspect of loss of repeat integrity. So what about tissue-specific gene expression? I told you that it also targets genes. Maybe actually the phenotypes, maybe the aging phenotype is linked to gene expression. So to do this, we had to isolate a tissue, and we chose muscle. So you can isolate muscles from worms or different tissues by using fluorescent markers and then sorting those cells, because actually there is no tissue culture for worms. It's all in vivo. But we could isolate a unique myosin, muscle-specific myosin, positive cells. These are the body wall muscles, and do RNA-seq, tissue-specific RNA-seq in both wild type, the double mutant, and the single mutants. In the double mutant lacking H3K9, we see about 310 tissue-specific genes derepressed. In MET2, almost all of them are derepressed, so there's nothing attributed to anything else. And SET25, which is the trimethyltransferase, doesn't repress anything. That's, again, proof that the dimethyl compensates. So what are those genes? So we map them back to where they are normally expressed. So this is called a tissue atlas. You're probably familiar with it. Each bar here is a gene that's derepressed in the MET2 versus wild type, at least twofold. And then here are all the tissues where gene expression has been expressed. And it's really striking, the majority of the genes that are derepressed, or the only tissue that's really, really overrepresented, is the germline. So the genes that are involved in maintaining totipotency are repressed by K9 methylation in differentiated tissues. It's not just a germline to soma transition that is mediated by MET2. It's actually because we showed that actually in tissue, in differentiated muscle, if you downregulate MET2 in terminally differentiated muscle, you derepress the same set of genes. So it's a continual maintenance of repression of germline genes in differentiated tissues. Another interesting phenotype of loss of canine methylation is that muscle integrity, so these are is lost. So these are the sarcomeres of the body wall muscle, which is the major muscle of the worm. And you see these nice actin-myosin structures. And in the MET2 mutant, this is an actin staining in a young adult. We see all kinds of deformations, and the worms become less mobile as they age earlier as well. So it's a really striking premature aging phenotype. So these two mediators of genome stability or genome organization, of repression, of segregation of heterochromatin from euchromatin, have specific jobs. They are definitely on tissue-specific genes, they work together, but in fact MET2 is the fallback and the essential mediator of gene repression, and they have physiologically important roles. Now, the phenotypes are really distinct because MET2 mediates all those phenotypes I just went through with you, the genome instability, the early aging, the loss of germline, et cetera. And SET25 has none of that except RT, retrotransposition, loss of stress-induced heritable silencing, transgenerational silencing, and it seems to be compensated to a large part by argonauts and RNA degradation. Now, this should be to you reminiscent of things that we know about humans, or humans, that actually in aging we lose heterochromatin stability, or the repression, the organization of repeats at the nuclear lamina, that lines which are and sign elements that are never expressed in early life start being expressed completely without a disease other than aging, if aging is a disease. And this is a quantitation of young versus old across all kinds of tissues. Also in cancer, we see that satellites in particular are de-repressed. In 80% of the epithelial tumors, we can detect major satellite 1 expression due to loss of proper repression in human tumors. So this is very much reminiscent of what happens in worms. So those are, for me, that underscores the importance of organizing this dark side of our genome and keeping it repressed. The question is, does it really matter? And the only way to answer that question, I mean, it does matter if you say, well, we see, a global aging, instability, et cetera. But can we trace specific diseases to specific loss of this spatial organization? So the question is there a nuclear scaffold? Is there something that organizes this genome and the repressed domains, separating them from wild type? And years ago, this was the lab I did my postdoc in, actually. By extracting nuclei, one was looking for some sort of organizational element in the nucleus. The only thing that came out were actually the lamins. There's no actin, no microtubules. There is some RNA-bound proteins that precipitate even when histones are extracted. But really, it was just lamin A, lamin B1 and B2, and lamin C, the intermediate filament family, that was somehow structural. It forms this meshwork that, of course, as you know, encircles the nucleus, and all the nuclear pores are embedded in this. And it is sort of the scaffold of the nucleus. Now, it's not so simple. It's not just the lamin proteins. In fact, with time, we know that there are at least 10 conserved. Some of the membrane proteins, lamin-associated proteins, are membrane spanning, like emerin, a lamin B receptor, MAN1. And they interact with proteins that interact with DNA. But most of them don't have direct interaction other than the lamin B receptor. And then there's the sun domain protein, which actually spans and interacts with the actin filaments in the cytoskeleton, so that you can have actually crosstalk all the way from cytoskeleton mechanical stress to the genome. So the question that was raised, of course, was what's the importance of this with the H3K9 methylation? Do they cooperate? How do they interact? Does the lamin bind DNA? Does it bind H3K9 methylation, et cetera? So the best evidence of or a way to map this was through an assay that Bas van Steensel developed in the Netherlands, where he fused DAM-ID, which is a bacterial methyltransferase that methylates A in the GATC in bacteria. But if you introduce it in the humans, it has the same activity, of course. If it's diffusing all the way through the nucleus, it'll methylate every GATC it finds or has access to. If you fuse it to lamin or emerin, then it will only methylate the DNA that is juxtaposed to the envelope. So he has done that in many conditions, many cells, and the picture that we get here in pink are the sequences that are really associated with the nuclear envelope. They're clearly enriched in lines and LADs, and they're late replicating, depleted for genes, and the green, which are the non-lamin associated, are enriched in genes and allele elements and a compartment of TADs. So this just confirms what we see in the microscope. But there was more. Very interestingly, you can correlate that with R and G banding if you're interested in R and G banding, which were GC-rich, AT-rich, although the percentage of difference is very, very small. But it does translate into the banding pattern does translate into the same kind of separation into line-rich, paranuclear heterochromatin and sign-rich euchromatin. Now these do change, and not only do we get H3K9 methylation on the genes that have to be silenced in tissue-specific manner, as you differentiate ES cells to astrocytes, passing by neuronal precursors, you see that genes either gain lamin association or lose it. Those that seem to gain lamin association are stem cell genes, like I said, germline genes, and those that lose it might be neuronal specific genes. And in this way, he could actually quantify a number of genes that basically switch direction or association as cells differentiate. Now, this confirmed, again, a model that people had seen in so genome-wide, a model that people had seen with individual loci. Like this is from Wendy Bickmore's lab, watching a single gene shifting from a state where it's repressed at the edge of the nucleus, and this is the chromosomal territory, to the interface of the chromosomal territory when it's turned on. So the chromosome position doesn't change, but the activated locus does. Or work from my own lab in worms again, where we looked at undifferentiated nuclei and then what happened to tissue-specific gene as cells and tissues differentiate. And we found a random positioning, even genes that were on or off in the very early embryonic nuclei. But as tissues differentiated into terminally differentiated cells, inevitably, the repressed tissue-specific genes became perinuclear, the active ones became internal, and this was actually shown in my lab for about 7 different tissues. Housekeeping genes, interestingly, were random. So what you should take from that is that nuclear organization is largely transcription-driven or sequence-driven, not necessarily transcription-driven, and that heterochromatin tethering provides a polarization of chromosomes that then may be diagnostic of the cell. So now, does it matter? So the only way for a geneticist to probe this is to look for mutants, and the best example I know of are the laminopathies. So the lamin gene I've already introduced. It is actually the most frequently mutated gene in natural mutations in the human body. It's only 70 kilodaltons, the protein, but it harbors over 400 distinct point mutations that have been mapped in people with diseases and mapped to 14 different diseases. If we zoom in on this, they extend all along the gene, particularly those that affect muscle are found basically all along the gene. Those that affect aging tend to be C-terminal truncations. But there's also a cluster that affect adipose tissue and nerves in different sites. And I'm going to tell you a very brief story about one of 280 different mapped EDMD mutations, just because it proves a point. Emery-Dreifuss muscular dystrophy is this family of human diseases. It's autosomal dominant, so a single point mutation in a lamin A gene, a single of the 2 lamin A genes will lead to this late onset, or let's say adolescent onset joint deformity from early childhood to teenage years, slowly progressive muscle weakness and wasting, and then heart problems by adulthood, and most of them die before they're 30 with cardiac defects and abnormal heart. They have increased risk of stroke and sudden death. So it is, you could say, an orphan disease or a rare disease. On the other hand, if you realize how many mutations there are that lead to muscular dystrophies in lamin, it no longer seems so rare. So we introduced it into worms. Actually, there are no really good mouse models for many of these mutations. But you see how conserved the lamin is. It was very easy. We could introduce an ectopic copy fused to GFP with the identical point mutation so that it was expressed on top of the normal complement of lamin, which would be the situation in a human. And in a swimming assay, which you've seen the muscles of the worm, if you put the worm in water, it flips its head. It loses its ability to swim, could be restored by countering this aberrant lamin. And when we examined again by phalloidin staining, we see that this striated muscle phenotype was really perturbed. Now we looked at gene expression, and actually we knew, as I've just told you, that when tissue-specific genes are expressed, and this is the myo3 promoter, the one I even mentioned before, it shifts to the inside of the nucleus because it's a tissue-specific gene expressed in most muscle cells. This is wild type. And in the mutant, it stayed at the nuclear envelope. So this was a gain of function phenotype where rather than shifting like it should, it remained attached. So this point mutation in the lamin was sequestering DNA. Now, we had already identified anchors for H3K9 methylation at the nuclear envelope. It's a protein called CEC4, which makes a ring around the nucleus that has a chromodomain that binds actually H3K9 and tethers H3K9 marked chromatin at the periphery. And so we wondered if the lamin mutation was either mimicking this or if the lamin mutation, or antagonizing this, since the lamin mutation was having hypersequestration and this was having loss. So we looked in the CEC4 mutant in wild type. This is the myo3 promoter on an array integrated in a genome. It's internal in the nucleus. In the CEC4 mutant, it's still internal because there's no reason for it not to be. In the lamin gain-of-function mutant, it's at the periphery, or at least 50% of it is. And in the double mutant now, we release it. So that was really neat. You know, we have a gain function. We could counter it with another anchor, suggesting that somehow there was cooperation between the lamin and the CEC4, both of them at the nuclear rim. And so now we ask the question, if we release that, you know, is just position enough to restore the normal muscle function? So we looked at the swimming assay again, and the CEC4, of course, has no effect because it has no effect on the position of genes in normal. But in the double mutant, we see the drop in swimming. This is the swimming assay in a bell curve. Swimming assay, the mobility drops, and it is compensated, not completely, but quite efficiently by the CEC4 mutant. Now, was this only unique to this gene? Oh, and also, amazingly, we could restore the striated muscle. This is the striated muscle in the lamin mutant, defective, and the not complete restoration, but almost full restoration. So was this just, you know, a unique lucky gene that we looked at, myo3? So we did a genome-wide mapping of the position of sequences to with this DAM ID that I introduced with the nuclear envelope. And the blue is the gain-of-function lamin mutant, and you see this higher peaks beyond the wild type. So each one of these is a chromosome. And you see that there really were a lot of sites that were then more sequestered at the periphery than in wild type, in the mutant. We pooled those into areas and asked, okay, so these are the bind with log2 enrichment. You see a strong enrichment at the periphery. Nothing becomes more released. We asked, what's there? I mean, they're genes. Is there anything specific about these genes? You know, we're talking about quite a bit of the genome. And we looked for transcription factors. Well, first of all, we looked for repeats, and we looked for H3K9 methylation, but they were not enriched in those regions. So we then looked, were there any genes that were particularly important there? And to identify the genes, of course, you can look at lots of genes, but you know, if you've got 300 gene names, it doesn't actually tell you that much. We looked at the transcription factors that might be co-regulating, enriched in those genes that were sequestered, and it was strongly enriched in the two E2F alleles. So E2F is a transcription factor that's bound by RB, regulated by RB, and RB is known to bind lamin. So what is RB? RB is an inhibitor that inhibits E2F, and E2F is famous for its role in the G1S transition, but it also regulates other factors. So A-type lamins were known to regulate RB protein function by promoting subnuclear localization. And this was published already in 2004. But what was published also published early, was that emerin at the nuclear envelope also disrupts the RB and MyoD pathways during muscle regeneration. So what we found with our very long, windy road from the point mutation in lamin was that, in fact, we were affecting a class of genes that are regulated by RB. Now, there's a massive number of alleles. The question is, how many of them actually, in this way, probably very few. We introduced actually 10 different alleles, and actually only 2 of them seem to be gain of sequestration. Many of them lost binding. So I'm not saying that they all act in the same way. They certainly don't. But the take-home is that nuclear organization, or lamin integrity and the sequestration of genes at the nuclear periphery, if you compromise this, you can drive disease, and chromatin positioning can misregulate differentiated cell pathways. And it's not only in muscle or adipocyte tissues, which are affected by lamin mutations, but also it's been reported for hematopoietic and other pathways. So I think this is actually this global organization of the genome is an important player in genome integrity and cell type, cell fate. Now I'm just going to skip about aging and cancer. So what my main point is that actually you can go from the very basic status of basic chromatin to the global organization of chromosomes and actually interpret cell fate. Now the question is, can we use it in any way that's meaningful, rather than just the cell biologist, amusing herself? So the question was, now in my old age, I'm in a cancer institute, running a cancer institute. We wanted to know if we could automate nuclear imaging for diagnostics and use machine learning to use nuclear image analysis to diagnose, in a sense, to augment the power of H&N-E staining. And so we asked the question, can you infer, based on chromatin organization, the status of, let's say, checkpoint inhibitor sensitive or insensitive melanoma? Can we link the melanoma phenotype to chromatin features of the tumor cells or of T cells? Can chromatin features actually be monitored in tissue samples sufficiently to guide diagnostics? And this is the work of a postdoc at our new Agora Center. So a tumor is not just tumor cells. A tumor is a whole group of stromal cells, which are very largely immune cells, and then epithelial cells, vesicles, etc. And so in any tissue section, you have all of these. So what we're doing is just staining with DAPI on top of an H&E section, analyzing the DAPI in an automated manner with image processing, using then machine learning to analyze the shape of the nucleus, the condensation level of the heterochromatin, the orientation, the curvature of the nucleus, the size, 200 different parameters, and then correlating with cell status. Of course, in initial state, we're also staining for markers of both the tumor and different immune cells, so that we can see whether we can correlate our DAPI staining with cell state. So actually, this we extract the feature. I'm not going to take you through all this, but I will jump ahead. We are able to differentiate based on a selected subpopulation of the DAPI staining characteristics, the other cells from the melanoma cells in a significant manner. And this is just a different portrayal of this. And then if we take the cancer cells versus the normal cells and resolve the heterogeneity in the tumor, we see that they fall into a number of classes, subclasses, and these subclusters of cells we could correlate with the different markers that we use to identify cells there. I won't take you through all of this, only to say, that we were able, or HITEM was able, with a random forest classifier, etc., to be able to separate SOX10 plus cells, those are the tumor cells, from the tumor-invading immune cells. These are activated T cells. Separate SOX10 cells from CD4 plus immune cells. Separate immune cells from the double plus immune cells, both CD4 and CD8 from unactivated immune cells, and separate CD8 alone from PD1. These are If you're not an immunologist, it doesn't mean anything to you, but there are different classes of T cell activation. In short, it works. So my message is, nuclear organization is not only important for maintaining genome stability, maintaining tissue differentiation, but it can be used in a very, very basic, almost naive way. Well, naive. Machine learning-based tool to actually characterize tissues. And if we use this then and correlate it with higher dimension, do a higher dimension correlation with, of course, multiomics and other information, we think that we may end up with a nuclear organization-driven diagnostics that will outperform our current pathology tools. So differentiation correlates with nuclear organization changes, and I hope I've convinced you that it's interesting. This is our new institute. These are the Alps beyond the Lake of Geneva. Haitem Shaban is the postdoc that did all that analysis, and this is my former lab on our last lab hike in Switzerland. Thank you. And I ran over, sorry.
So
maybe we could ask questions during the coffee break or something, unless there's something pressing.
I think we have time for a few questions, if there's any in the audience. Otherwise, I'll start with one. So for the first part of your talk, all the repeat expression, I found that very, very interesting. There's two questions I have. In cancer, we use a lot of HDAC inhibitors and demethylase agents that act through the repression of repeats. Do you think we should be taking into account the DNA damage that because in a side-off effect?
Well, I think you want DNA damage.
Oh, for the cancer
you're using
cells,
those
for sure,
to perturb the cancer cells and express neoantigens and so forth. So, I would worry about the DNA damage you might be provoking in normal
That's exactly
dividing
my question.
cells.
Yeah, I don't think we are looking into that.
Fortunately, in normal cells, first of all, most of our cells aren't dividing, so you're not getting replication stress. But of course, in the blood cells and so forth, you're going to be causing damage. I think that used to be my message, like if you're BRCA minus, don't touch the H3K9 inhibitors,
Exactly.
But might kill you. But I think the fact is, there are lots of backup mechanisms. So especially if you have a specific, like G9A inhibitor or a specific inhibitor, it's a lot better than a pan inhibitor. Same thing for HDACs. I mean, the worst thing are the pan inhibitors because they're going to be provoking, too much derepression.
Yeah.
And I think, depending on the tumor, it can be useful to have a specific, like G9A inhibitors I know are effective in certain tumors. So I think it's just a question of detail.
And I have a second follow-up question, if there's no questions in the room. Yes, there. So I won't monopolize. Yeah.
For me, a little bit surprised that double mutant still survive. So it means some other epigenetic marker can compensate for H3K9 maturation function.
So of course, everybody asks me, does K27 go up? And it doesn't, actually, not in a massive way. I think that in worms, they have a very efficient small RNA pathway that degrades promiscuous transcripts, and that's part of the repression, and we find a very strong synthetic lethality with an Argonaute mutant with MET2, stronger than CET25. So I think there are compensatory mechanisms that somehow allow those worms to survive. Remember, they're sterile, so they're not very healthy. But they do differentiate. and for me, what that tells me, so worms don't have somatic stem cells. They just differentiate all their somatic cells. What that tells me is that actually point mutations, even insertions, deletions, and instability in somatic tissues don't interfere with the forward differentiation program. Although you see premature aging, you see degeneration of differentiated tissues. I think the forward differentiation pathways are driven by transcription factors like dominoes. You know, you just titch one and then you differentiate. And that's why in worms, you're not maintaining stem cell pools except in the germline, and therefore you can differentiate. That's my interpretation.
Yes, go ahead. Oh, I'm so sorry.
I haven't
coming.
got just for those that are online. So the differential gene analysis in the mutant worms that don't have H3K9 methylation, that was all done in muscle, is that right? Just wondering if you've done that in other tissues or cell types and if you saw a similar pattern.
Yeah,
And
we
the
did.
follow-up is if you've done that in, I don't know if mice, if there are equivalent experiments have been done in mouse mutants as well, or if that's possible.
So we did it in, we compared muscle with hypoderm, which is a skin cell, and a gut. And in all of them, germline genes are de-repressed when you get rid of K9-methyl. And then, so there are 3 classes of genes. So in the embryo, the genes that are K9-methyl repressed are different than in differentiated tissues. If we compare 2 differentiated tissues, there are some genes that are just de-repressed in muscle, some just de-repressed in gut or skin, and a large number overlap. And many of those that overlap are the germline genes. We did ask the question, why are there different genes? You know, they all have K9-methyl, you know, like all the neural genes are K9-methylated in both tissue, both non-neural tissues. It's the availability of transcription factors that determine whether, so loss of K9-methyl is not sufficient to turn a gene on. You have to lose the repressive mark and have the appropriate transcription factor. So that's probably also why it's only like 300, 350 genes that significantly get up, because the transcription factor has to be present.
But something has to hit the transcription factor.
Something has to what?
Something has to hit the, I mean, it has to be upregulated.
No, it doesn't have to be upregulated.
But if you go from stem cell state into differentiated cell.
I mean, as you differentiate. Yeah. No, I mean, there is a cascade of transcription factors that are, I think, is just, you know, it's like a transcription factor, you know, you activate a transcription factor that activates a transcription factor, et cetera. But, so transcription factors are reused. So, in different tissues to regulate different genes. And the availability of the promoter is regulated by epigenetic marks.
Yeah, we can.
I think
I'm thinking it going from a stem cell, it's difficult what's the hen and the egg, what hits the button when you have this stem cell. If you have a metabolic signal, for instance, deciding to induce differentiation, and then is it the epigenetics or inducing, and do you have to have epigenetics hitting, inducing the transcription factor?
So you know, there are only, I mean, the number of transcription factors that we have and the number of differentiated cells we have, there are not enough transcription factors to account for all the differentiated phenotypes we actually have in our bodies. So factors have to be reused. And they're reused in different combinations in different tissues. So a specific factor may not be expressed in muscle, and then if you lose the K9-methyl, the gene won't turn on. But there are genes that are normally repressed only by the epigenetic mark. That's what I showed, because I actually, we actually turned off the K9 methyl, or the HMT, in differentiated tissue by Degron. And the same 350 genes came on. So these genes were absolutely being repressed by the methyl mark, okay? So it wasn't a question of turning on the transcription factor to turn on that gene. The transcription factor was in the cell. It was just not accessing the promoter. So this is a very, very fundamental question, actually. And that's what I think was the most important point of this paper that we published in 21, is that actually epigenetic sequestration is sufficient to repress genes. And keep them away from transcription factors that are actually in the cell.
I think in the interest of time, we'll cut the questions here, and then we'll have the whole day to keep asking questions. I do have several for you.
Thank you very much.